The headline on the other major news story today, to which we intend to devote some time, is very simple. Hello, Dolly.
In February of 1997, a Finn Dorset sheep named Dolly sent waves of future shock around the world.
The first living, breathing clone mammal.
It's possible we're seeing a scientific explosion comparable to the atom bomb or the moon rocket or DNA itself.
For many, it was a case of science gone too far.
Are we acting more like the creator than creatures? Are we trying to play the role of God in all this?
Predictions multiplied about just what this breakthrough would bring.
Soon it will be possible to give her thousands of absolutely identical sisters.
Animals could be cloned with human diseases and new therapies tested on them.
Endangered species can take heart.
This is not an elaborate, sophisticated technique. It means that any decent college or graduate school student could potentially clone a human being.
Whatever became of Dolly, and all that speculation about the brave new world she ushered in.
'97 was a long time ago, but I remember my wife and I were in a meeting, just normal grad students with no money, broke, completely broke, but we just so happened that we were doing similar work that Ian Wilmot and Keith Campbell were doing at the Rosalind Institute. And automatically, our salary, I think it was increased 5 times the day after Dolly was announced. So it was one of those things that the tide raises all boats, and we're forever grateful to Ian and to Keith for doing that. And I'm just gonna start by remembering Ian. He passed away about 10 days ago, or a couple weeks ago. He studied at the University of Nottingham in Animal Science Department, and then he went to Cambridge, where he did his PhD with Professor Palch. His specialty at the time was cryopreservation, and so Ian was the first one to get a calf, actually, from a frozen and thawed embryo. Then he became part of the Rosalind Institute. At one point, he was the director of the institute, and then after Dolly, his life completely changed, and he was traveling the world. He just stepped down and was the faculty member until 2005, when University of Edinburgh created this Regenerative Medicine Institute, and he was the director of the institute. And retired in 2012, and we had the opportunity to talk to him a few years ago. I think it was 2018, and we did a nice interview. I'll show you just a little bit later. So these are the 4 stages in a relationship with cloning, particularly my relationship with cloning. In the 1990s, it was a total intrigue. I guess we were mystified by the fact that you can take any cell in the body and put it into an oocyte and then put it into an animal uterus, and you get something. So the first thing we got were fetuses from fibroblasts, from fetal fibroblasts. That was back in 1996, late 95. I remember the cows aborted the fetuses, and they send us the fetuses. We would do the embryos in Massachusetts, ship them by FedEx to Texas, and then they would transfer them into cows. So they sent us back a container full of aborted fetuses, and I was devastated. And my advisor looked at me and said, well, if this is real, all you had to do is keep at it. And of course, we were talking with Ian and Keith Campbell, and we knew that this was real. So we were mystified. But then the next challenge was to figure out the mechanism. And so we were getting grants and trying to get private funds to understand how a somatic cell can go back in time and back in differentiation. And it was 2008, and ICCR meeting in Canada, when Yamanaka announced that he had done it without any need for an oocyte, right? So that was my reaction when that happened, because I was still in love with, of course, with nuclear transfer, and I wanted to keep working on it. So we needed to find a different angle to figure out the mechanism and make sure that it was still current and the funding agencies were willing to pay for it. So This is what I'm doing these days. I'm just going along and trying to, with a lot of less funding than before, but we're still making some progress. So the 4 milestones I want to talk about are 1952, Brigg and Kin in Philadelphia. They were the first ones to show that you can do this in frog. Then it was Rana. They did it with Rana blastomeres. In '62 was John Gordon doing it with Sinopus. I'll walk you through it. So this is 52, Lankino Hospital in Philadelphia, where Brigg and Kin did a transplantation for the first time. They took blastomeres from an embryo and put it into a recipient egg from a the same species, but they got tadpoles, and then they got adults. So these guys work with Rana pippins, but you had to take their word for it. There were not genetic markers. There was not such a thing as PCR or things that they can test for to make sure that this came from a different animal. And that's when you have the elegant experiment, as it's called. This is Gordon, that went on to win the Nobel Prize in 2012. He took cells from the intestine of a frog and transplanted it into an egg, and sure enough, he got development. So the few things that he did different, well, he inactivated DNA of the recipient egg with just UV light, and so this was easy, no manipulation needed. The second thing he did is he would just inject cells that were coming with a different phenotype. So it was the same species, but a different phenotype. So they found in their lab, they found that there's a mutant that had only one nucleoli, when the wild type also always had 2 nucleoli. So this is what actually made the world believe that the actual experiment did work. It was not a made up. 1996, this is pre-Dolly, but the Rosalind Institute was sponsored by a private company trying to produce recombinant proteins in milk in sheep. And so they used embryonic cells isolated from a blastocyst that were plated in a petri dish, and they were cultured, and then without any particular culture media that prevent differentiation, those cells turn into what it looks like fibroblasts. Now those fibroblasts then were used for nuclear transfer, and they were able to produce offspring. And so these are sheep that were born, and I can't remember, they gave them some special names, but the breakthrough was evident, so that's why they got a paper in Nature, and of course the '97 paper was with cells from the mammary gland of an adult animal. I think it was a 6-year-old donor. They have that cell line frozen in the lab. There was a lot of skepticism in the US. There were letters to Science magazine saying that it was it may have been a mistake. The cell lines actually may have been embryonic cells. These are the critics, right? But they took it in stride. These people were convinced, and we were all convinced that this was working. So what did we do during the 2000s? Well, we cleaned the heck out of every single species that you could. So replicated the experiment multiple times. More than 20 different species now have been cloned, from the wolf to, I don't know, cats and dogs and you name it. So where are we now? Well, depends how I look at things. So what I did in preparation for this talk, I did an informal survey with colleagues that are still working in the field. A lot of people left after Yamanaka published the IPSL. They said, well, we don't need cloning anymore, and we already cloned all these species, so let's not worry about it. But some embryologists like me decided to keep working in the field. And so I interviewed about 8 different colleagues, and I asked them 3 questions. And the questions are, do you still use cloning? What is your current efficiency compared to the efficiency that this person had published before? And what was the breakthrough since '97 that has helped you the most? So since Dolly was announced, was there anything in the field that oh wow, eureka, we found a way to improve the efficiency. So why do you still use cloning? People are cloning cows and cloning pigs that are genetically superior. So basically what you're doing is horizontal dissemination of the genome. And this is without any genetic modification, and there's a lot of money to be made, especially when you're trying to create founder animals. For example, in cows, if you have a very high value animal, you can only produce X amount of embryos a year from superovulation or OPU, or ovum pickup. So what you do is you just clone that animal, and then the female will then you have 10 copies of the female, so you make 10 times more money than by having only one. There's a lot of people, and actually there's this NIH center in Missouri that is run by Randy Prater, that all they do is create animal models for human disease. So they're modifying mini pigs, that they have mini pigs that have no immune system. They have mini pigs that are for a study of cholesterol. They're doing one for Parkinson's disease, all kinds of models. Not only that, there is a company in Minnesota that is trying to create organs in pigs, and for that they have to modify the pigs to allow for the organs to grow. Sport animal, big business. Polo horses are being cloned, and there's a lot of money to be made there. Companion animals. I interview Dave Favor. Dave Favor is the director. Well, it's the president and CEO of a company in Iowa, Transova Genetics. Currently, they have all the technology for cloning animals. And they actually license out pets because he didn't like the idea that he didn't want to be associated with something that could be controversial. There are so many animals that you could adopt that he just let it out, and another company took over. And then he regretted that because Barbara Streisand decided to clone her dog, and she got these 3 puppies, and the market exploded. So they're making a lot of money out of cloning dogs and cats, I should say. Rescue endangered species. There are many efforts around the world to work with. One I'm aware of, there are many, but one is the Bangtang, for example, in Thailand. They're making an effort to reintroduce different genetic variation so that the herds can actually be improved. And they're using cloning for that, but there are many other examples. And germplasm repository, and that's where we come in. We're currently funded by NIH to do a repository of germplasm in zebrafish. Zebrafish is a species that is being used, is actually the fastest growing animal model for a study of human disease. We share 70% of the genome with them. And there are about the last count, I think it was 47,000 different mutant strains of zebrafish that you can study. And what happened is you create those mutants and you do your paper, you publish well, or you find your drugs that you're looking for, and then what do? There's no way you can freeze the embryos. The embryos are too big. There's about 100 microns in embryo. So what people are doing, they're freezing sperm. And then when you want to get somebody, any of you wants to study a disease, that you go to the browser at Oregon. They have the Zebrafish International Resource Center is in Oregon. And you just go there and look for the genes you're looking for and put a request. And what you get about 2 or 3 months later, maybe sooner than that, you get a bunch of 200 embryos that were eggs fertilized with the mutant sperm. But what you're getting is just half of the genome. So you're not getting the whole genome because you need to use wild-type females to use the sperm. So we propose to do the germplasm repository for zebrafish using diploid cells, and then we just clone from them. So what is the current efficiency? That was a big question. And some of them were commercial entities that were not very willing to share, but after talking to them, they just share with me. So we're stuck in the 90s. Okay, nothing has changed much. As you can see here, this is with exception of zebrafish that is somewhere in there. This is the efficiency of animals born healthy out of the females that receive a clone. So you have sheep, goat, bovine, pig. Those are the domestic species that we know most about embryology. We're in the barely 10%, maybe 20%, depending on the animal. Actually, there is variation between genomes. So you could have Holstein animals that one only give you 1% efficiency and another will make in the 20% efficiency. But and overall, we're stuck in that efficiency. So what is the breakthrough since '97 that has helped you the most? So this is a recent conversation I had with them, and there are so many papers that are showing all these epigenetic modifiers that can be used to help cloning. Well, the answer was consistent. Nothing big. It was just little tweaks in here and there and improving maturation of the eggs or culture media, but nothing that you say, okay, if I put this in the cells before cloning or during cloning, I'm going to get the same efficiency as an embryo that was fertilized. So, on last count, about 3,400 papers in PubMed. Do we know anything about the mechanism? Okay. We don't know anything about the mechanism. Otherwise, well, we know a little bit, I would say. I mean, I can explain it, but rough details, but nothing that you could. For example, when you make iPS cells, people are still trying to figure out the mechanism. Yeah, you know you have to use these pioneering transcription factors or epigenetic modifiers, but exactly the mechanism is hard to pinpoint. So the mentions that I want to talk briefly are these 4 papers that came out at different times. The first one is from Japan. It's an article that shows that the use of trichostatin A. Trichostatin A is a small molecule that will actually increase histone acetylation because it's a histone deacetylase inhibitor, and also decrease DNA methylation. I guess summarizing and generalizing what the effect is, you open up the chromatin and you try to remove the repressive marks and let the oocyte do whatever the oocyte has to do to produce a clone. So this is, the efficiency was increased from 0.1%. Cloning mice is very hard. Let's start with that. Cloning mice in the hands of the experts like Inoue or Wakayama's lab, Ogura's lab. These are Japanese researchers that are top-notch in cloning mouse. Their efficiency is about 2-3% in general, unless they do something unique. And in this case, they use trichostatin A after injecting the nucleus of the donor cell into the oocyte. There was an incubation period about 10 hours. And then after that, you release the embryo, and it goes on to produce pups. The other article that was interesting came from Ogura's lab and Inoue, and they've been working together for decades. What they show is that X-cyst expression, so the gene that is expressed by X chromosome, if you just block the expression of X-cyst, you can actually improve the efficiency of cloning. And this is an experiment they did. The knockout the X-cyst from one of the 2 chromosomes, and they got an increase in efficiency. I want to call your attention to the 2 donor cell lines, cumulus cells and Sertoli cells. If you remember a little bit about histology back then, these 2 cells are almost homologs, right? So in the male, Sertoli cells will respond to FSH, and they're in the seminiferous tubes. And cumulus cells are inside the follicle in the ovary and also respond to FSH. And they have a very intimate relationship with the gametes, right? Sertoli cells is where the spermatozoids are made, and they're completely connected with the sperm until spermiation, when the sperm releases from the Sertoli cell. And cumulus cells are connected to the oocyte through gap junctions. So there's something going on. These are the most efficient somatic cells tested. And the rest, yeah, you can clone from other cells, but these are the 2 best. 2014, this is to me the breakthrough. So this is Zhang from Harvard, Yizhen, and his postdoc Matoba. What they did is they did a very simple experiment where they sequence RNA, they did RNA sequencing in embryos before and after genome activation in the mouse, okay? And then compare the fertilized ones with the nuclear transfer ones. And they realized that when they map the chromosomal regions where those genes were either expressed or not expressed, they ended up with a pattern of histones that were modifications. That was H3K9 was the methylation, 3-methylation was the one that it was persistent in the clones. And so this is a highly repressive mark. And what they found is that in the clones, there were genes that shouldn't be turned on, but they didn't. And those are what they call resistant to reprogramming. And genes that should have been shut down because they were somatic genes, and they were not. They were still active. So when they put the histone demethylates in it, like they injected directly, they injected mRNA for histone, demethylates. What they got is increase in efficiency that was then replicated in human. So they produced human embryonic stem cells using cloning, and they also increased the efficiency using this. And the final paper came from the lab of John Gordon, the guy that cloned the frog in the 60s. He's still working in his lab today, and he also modified a histone residue, and this histone residue is actually not a repressive mark, but a permissive mark, and is the one that gives somatic cells their expression of the genes that are specific for fibroblasts or cartilage or anything. So they remove the memory of those cells by changing this mark. And now, do these findings apply to all species? Unfortunately not. And this is one of the things that we have. Actually, I really liked yesterday, I can't remember who presented the data on the skin of the, I think, tilapia. And actually, I'd like to see, yes, I really like that because you show us negative data. And not everybody wants to do that. And so that's a problem in our field as well. We try many different things, and we don't publish it. We just put it under the rug, move on, because everybody wants to publish positive results. Everybody wants a high-profile paper. So if conferences are a good place. This is where you can actually find out what's really going on. But not everybody can travel, especially during COVID. So what are the things that we know are true in cloning, most species in cloning? Cell cycle compatibility. You can clone from almost any cell type. This is something I'm not going to elaborate much, but it was also consistent through the colleagues that I interviewed with. The cells, as soon as you take the cells, whatever cell type you choose, it could be mesenchymal stem cells, it could be skin cells, as soon as you put them in culture, in vitro, everything changes. And no rhyme or reason. For example, in cows, you can have the same animal, you get 2 skin biopsies, you make your culture of fibroblasts, and you get 0 pregnancies with one, and the other one will give you 20% efficiency. Same cell type, all the markers for fibroblasts. And so this is something that Surani used to call it methylation drifts, but I think it's more than DNA methylation, otherwise we could have fixed that. Cross species doesn't work. This is from the mouth of somebody that was also, I was passionate about cross species. I mean, we cross bovine cells with monkey. We put monkey cells into a bovine oocyte, always with the hope that we could actually use this for rescuing endangered species at some point. And it doesn't work. The species have to be really closely related. So we got it to work between bovine oocytes and the gaur, and that is a very, very close cousin. And some people got it in different subspecies between in sheep. Others have done similar work in feline. But if you go far, like some people are injecting nuclei from the holy mammoths into the mouse eggs, and the DNA doesn't go anywhere. Of course, the DNA is also fragmented. But So the method used for activation is the key. We don't have sperm, right? We don't use sperm. And sperm is what triggers activation in the eggs. And healthy clones will age normally. Because of Dolly was cloned from a 6-year-old animal, people thought that it would die sooner. Indeed, it died sooner, but it wasn't because of cloning. I'll show you some data on that. So cell cycle is important, I said, and usually we all know when the oocyte is ovulated, it's stuck, right, at metaphase 2, meiosis. So if you look at the mature oocyte, under the microscope, you can pretty much see the spindle, and you can see all the chromosomes lined up. If you were to fertilize that, sperms come in, and the egg, and then extrude the second polar body, and you get haploid DNA content, and moves on. In cloning, we don't have sperm. I told you, you have to remove the DNA of the egg, and what you bring in has to be the same, at the same point in the cell cycle. So we all use G0 or G1 cells, and that seems to work fine. Donor cell type, people have tried multiple things like the natural killer cells, keratinocytes, embryonic stem cells, iPS cells. All cells will work at different efficiencies, all right? But they will work. So you can clone from almost any cell type. And this is the experiment I want to show a little bit more detail. Look at the publication. It's 2001. This is like a 2 decades old paper. But Jean-Pierre Ozel was working in INRA in France, and he was working with rabbits. And he was trying to parthenogenetically activate oocytes. And he said, well, we were all aware of imprinting, right? So you can't get a partheno to go on and make a healthy animal. But he wanted to see how far he could push it. And so what he would take embryos, transfer them into a recipient, and compare at 11.5 day of pregnancy, take them out. So you have here the fertilized group, and this is what they look like, right? The brain is developing normally here. And then these are all parthenos that he activated using electrical pulses. So he would put this unfertilized egg, inside a chamber and zap it with voltage. And look at the different voltages. So here he used 4 pulses, 4 pulses, 65 volts, 32 minutes apart. Here 8, 65, 8 minutes apart. So he was changing the parameters slowly and transferring them into the uterus and then taking it back at day 11. And look at this, what happened. With 4 pulses, 75 volts, 8 minutes apart, you can get almost normal fetuses. So just by changing the calcium spikes inside the egg, calcium release, you get all these changes in development, and it's fascinating. So the conclusions is small changes in calcium signal dynamics have a strong impact in both in the immediate response and, of course, long-term post-implantation. The issue about cloning, clone animals are not healthy. I'm going to recognize that most of the animals that are born alive, they may have issues at birth, and they may have problems with temperature, almost like a preterm birth. You could have the need for using oxygen and also to regulate glucose for the first 48 hours. But in general, they go on and thrive. The ones that we lose, those we do lose a lot, and they are mostly associated with placentation problems. So the placenta is enlarged, you have large placentomes, and it actually puts at risk the life of the recipient animal as well. Now the lifespan was something that we got involved to, but I'm going to show you a picture of Keith Campbell. This is Keith Campbell in the backyard of his house, fishing. And this was the answer when people say, well, really, Dolly, why did Dolly die so soon? Well, Dolly was actually sacrificed. She had lung issues, an infection in the lungs. And then when they did the necropsy, of course, they found arthritis. Like if you look hard, you can find something. So Keith decided to take the issue in his own hands and went back to the same cell line that Dolly was cloned from, and he added some other fibroblasts from adult sheep, And he said, well, we'll do the ultimate experiment. We just clone them and just wait until they drop dead and see how long they live, right? So these are the pictures of sheep that were euthanized when they were about 12 years old, 11, 12 years old, and they have no problems. They were absolutely normal. And some, I think 3 or 4 in there are clones of Dolly sheep. Unfortunately, before this paper was published, Keith passed away. Things I got wrong. If you look at some of the things that I said 20 years ago, I would say the oocyte can take care of everything. Once you put in nuclei inside the oocyte, it can take care of anything. They will turn it back into an embryo, and it's going to have a pretty healthy animal. And I was wrong. Of course, I was wrong. This is the other thing I said. In a matter of hours, somatic nuclei is turned into an embryonic one. That's totally wrong. And talking to a friend, as he works at the Max Planck Institute, Michele Boiani, and I'm going to quote him because this is exactly Michele. And this is what he told me. The reprogramming process is not over within a few hours after SCNT, but it unfolds during multiple cell cycles. True. This suggests that reprogramming has a passive component, and things can take place more actively because the molecular machinery simply hasn't been put in place by nature. And that is absolutely true. I mean, you have an oocyte that is waiting for a gamete, is waiting for a sperm. And a sperm has been, for months, or for not months, but weeks, have been produced in the testes, and there have been changes in the chromatin. When you throw in a somatic cell, things don't go very well. 4 things that we keep doing and are not helping. Okay, so this is for anybody that wants to get into the field. We're wishing that a single gene, a single molecule will solve all the problems. And I think this is true for other things that we work in biology. We do a lot of time lab analysis, but we don't have reliable endpoints. So in a species like the bovine or the pig, we do a lot of in vitro experiments, and we look at the embryos for the first few days until they reach blastocyst stage. And if we modify anything in the protocol, and our endpoints are blastocyst, no blastocyst, or how many cells in the blastocyst, or how many cells in the inner cell mass or the trophectoderm. That is old school. I mean, this is not helpful. We need to be much more sophisticated with the endpoints. And then we analyze single embryos at a low resolution. This is changing, but for most people, this is what they're doing day to day. And we increase the resolution by pooling embryos. And I got to tell you, the heterogeneity between clone embryos, even though they may look the same, they're absolutely different. So this is what you get when you pool embryos. You get a beautiful Parmesan cheese if you use fertilized embryos, and then you get a mess of all kinds of different cheeses, and then you try to find the answer why this is the way it is. But I think we're moving into the right direction with all the new technology we have available. There's a bright future out there. So we have access to low input, high resolution molecular analysis in real time, or as close to real time as possible. We have thousands of data sets from single cells or single embryos. Someone said this to me recently, but I should add that we are now in the era of artificial intelligence, and artificial intelligence will help us a lot, at least analyzing very complex data sets and very large data sets. And then the fun part would be just functionally validate the targets, and that's the fun part. So I'm going to acknowledge the people that I talked to for this presentation. So Michele Boiani from Max Planck, Randy Prater from Missouri, Teru Wakayama from Japan, Mark Maserati is Brazil, Dan Carlson is the company Recombinetics in Minnesota. Ecker Golf also works in pigs and is pretty much the same as Randy Prater, but in Germany. They're creating all these different animal models. Dave Faver, founder, CEO of and chairman of Transova Genetic. And Cesare Galli from Italy, he's the first person to clone a horse and he's still working very actively in the field. Now, I started with Ian Wilmot. I'm going to end up with Ian Wilmot. And I want to remind you, Ian is the person that did Dolly, or led the efforts for Dolly, and also traveled the world, collected honorary degrees and awards from every place you can think of. And we met, and we had a long conversation a few years back, and this is just the last part.
On my wife's gravestone, the last comment is, a friend to many. You know, that's after listing, me, the children, the grandchildren, a friend to many.
Well,
to something along those lines would be.
The video doesn't work.
I would be very
what
proud.
he's saying is, I asked the question and said, how would you like to be remembered? And he thought for a while, and then he said, the human traits. I kept on thinking, and he said, well, in my wife's gravestone, after she names me, my children, my grandchildren, she in the gravestone says, friends to many. And that was the way that he wanted to be remembered. And I'm thankful for, I guess, spent time with him throughout my career. He was a great guy. Anyway, questions if you have any. Thank you. Oh, here. Any questions?
Thank you, Dr. Sibeli, for a wonderful talk. Can you comment about the potential role of CRISPR in this field and how it may oppose or complement the work that you and your impressive collection of international colleagues are pursuing?
CRISPR is being used on a daily basis on all the pigs they're creating. And they are also, what they're doing is they do genetic modifications in the somatic cells, and then they use those cells to clone. Some other people are using CRISPR just in pigs as well, but direct injections, but a little trickier when you're trying to find if there is any off-target effects. But people have used it also in cows, and they're also using it to create animals like safer food and things like that. I think the FDA already approved one pig and one type of pig and one type of cow that had knocked out a couple genes, and they are actually allowed to enter the food chain. So things are happening in the food area and in biomedical field.
I wanted to ask you, why do you think cross-species doesn't work? What's the conundrum there?
Well, it's mostly related to enhancers. The genomes have their own, even though we may have similar genes, the way that when they're turned on and not, that's one problem. And then the other problem is mitochondrial incompatibility. So, yeah, that's the problem.
I also have a question. So, great talk. Thank you very much. So you talk a lot about efficiency of cloning, and that's as if that's kind of the major barrier. I'm more interested, I guess, I don't know that I'm not too familiar with the history of all, but what did cloning actually taught us about what happens naturally in fertilization, in that magical moment where we have reprogramming happening naturally, where a mature cell, right, two mature cells meet together and reprogram to start it all over again? Like, how did cloning help us understand that process? Oh yes, there are a few, I should have mentioned recent papers where we have, the royal we, collaborators have found genes. The one that come to mind is DUX1, is a gene that was found to be important for cloning, and then they realized it was probably the pioneer gene for doing genome activation. So for zygotic genome activation, you need DUX. It also helps in cloning a lot. So we're sort of validating things in cloning that then will be validated in normal development.
We're doing kind of the same thing with zebrafish. So that's a way to learn. Some always say it's too complicated. Why don't you just study fertilization more closely? Yeah, but I just want to find the mechanism.
More questions for Dr. Sibeli? All right, well, thank you very much.
Thank you.